So, first of all, would like to thank organizers, Delhi University, for giving me this interview. So, my topic is exploring the meronome of Erica papaya with potential first-order human gene targets. So now, as we live in the era of the home, like genome, proteome, transcriptome, methylome. So I touched one of the aspects of the total microRNA, that is the meronome of the known plant, that is the Erica papaya. It is a papaya that we eat, and whether it is having any first kingdom role in the human genes. So first of all, the studies center around hypothesis that we are what we eat. So ultimately, another and the Indian remedies work on the people's nature more. So ultimately, we are what we eat, and the question is, do plant-derived microRNAs have any role in regulation of the human gene expression in a particular what? So to address this question, the study was undertaken using the Erica papaya as a model plant. So as we all know, the Erica papaya plant falls under the and India is a major source for the production of the Erica papaya. And in terms of the nutraceutical value, it has all the nutrients like magnesium, iron, calcium, and other vitamins. And when I talk about the phytochemicals, also it is having a rich in phytochemicals that have the certain disease element. So I think all the parts of this papaya that are being used for the treatment of the certain diseases has a medicinal impact. So what we are interested in taking the microRNAs from this plant. As we all know, these microRNAs are non-coding RNAs and are between 18 to 22 proteins. And we aim this mix for the potent as a potent regulator of the gene expression is human. So basically, this is the biogenesis pathway of plant and animal microRNAs. And why we have taken these studies, right? So this area is very new. Globally, very few labs are working on this area. It was started in 2012 when first discovery of the miRNA 168 from rice in mammals, that is proven to regulate the certain human genes, right? And so on. And we started this work in 2017, and we could publish our first paper in 2019 that maximum acylicum, that is the tulsi that we eat, that found to have a multiple microRNAs that can regulate the certain human genes. So in the presentation, I have focused on two objectives that we did. First of all, one of it is that is the computationally we could identify certain novel microRNAs from papaya through the EST as a reference data. They express sequence type data, and the second is the sequencing total small RNA sequencing of the papaya plant, and to study their conserved and the novel microRNAs. So while in the first study, we have taken the model kingdom as a Fede plantae and the EST data set of Erica papaya. So from that, through the homologic searching and the several bioinformatics studies, we could identify some 9 novel microRNAs from this prediction. As you can see here, we have set the certain parameters to fetch this kind of microRNAs as the minimum length, maximum length, and the minimal folding energy and the adjusted minimal folding free energy. So these are the two key microRNAs, the CPA MIR 36290B and CPA MIR 6034. So these two key microRNAs from all the available EST data till now, they are evaluating, they are targeting 37 human targeted genes. And along with that, also we did the gene ontology studies on the basis of the annotation of these target genes. That is in terms of the role of the genes in the cell component, molecular function, and the biological process. Along with that, also we did some protein protein interaction studies and the network pathway analysis studies. So you can see here the protein protein interaction of the Erica papaya microRNA derived targets hemolyzer inhibitor genes. And these are the top hub nodes. So ultimately, from the studies, we can say that we found two novel MIRs targeting 37 genes in human, and ultimately these 37 targeted genes are out of the 18 genes involved in the predominant cancer pathways and the repeated activation. Our second objective, that is total small RNA sequencing of this plant and to study their genome. So for that we have isolated the RNA content from the plant, and through HiSeq NGS platform we could have the sequential data, and through the bioinformatics studies after doing some filtration and the other procedure, we found several high quality reads out of that. And we did certain bioinformatics studies, and that we have done some reference mapping with the available data, and from that we have so we did the last 10 studies and the mRNA studies. So from that we found 19 hits from that, and then we map against the genome, we found 49 novel microRNAs. So together these 9420 hits and the 1798 hits, after the second structure prediction, we found total of 49 novel microRNAs from this. These are the list of novel MIRs that we found. And again, when I talk about the annotation part, we did actually for the geogenesis, that is in terms of the cell component, molecular function, and the biological process. So this is the again gene network showing the association between the significantly related genes. So these are the list of top hub nodes located through the topological studies, and this is the phyllo tree for Erica papaya, which is of novel MIRs and the reported MIRs. So from these studies, we could identify 4 novel MIRs and their human gene targets that are being depicted by this circus plot. So ultimately, from the studies, we could generate the 30 million reads using the NGS technology, and we have identified 49 novel MIRs, and 4 of them were found to target almost 70% genes out of total predicted human targets. And the cross-freedom studies of this novel Erica papaya microRNAs will have the implication in disease management with the diabetes, cancer, mental illness, and the selected gene. So ultimately, from these studies, the key findings are, as I have depicted, we studied that it is not mixed by EST approach. We identified 9 potential microRNAs, out of which 2 mix are novel. And from EST approach, this mix plays a major role in the control process, disease association, and regulating the downstream signaling aspect via predominant pathways of cancer, and may have a functional impact on a human disease. From the second objective, we could identify the target genes constituted a significant role in the various downstream signaling pathways as W entry, NANO, MAPK, PRA, along with the pivot activation and ligand pathways. So these are the target genes which were involved in the various signaling pathways and acted as oncogenes. And finally, we also studied the common mix involved via EST approach, the genome-wide approach, small RNA sequencing approach, and from reported microRNAs deposited in the MIR-based repository. One of our key findings that CPA MIR390 is common in all of the 4 datasets, and it has a potential value in terms of the expression of the certain genes. So from these studies, we could publish 2 papers from this data, and now we are validating this target mix. We are making the gene construct, we are making the mimics of this microRNAs, and we are creating through the gene expression studies in terms of the luciferase gene reporter assay and the other assays. So thank you.
Any question from the audience? No. Thank you, Dr. Patel, for this nice presentation. Okay. So with this, we come to the end of this session.
May I request Professor Vinod Kumar to
He talked about osteoporosis, and he talked about a new term, immunoporosis, and that's very nice that your term has been accepted by the scientific community. And finally, Dr. Patel, he talked about the microRNA sequencing from the plants and the presence of common microRNAs between the plants and the human system, which also has a lot of potential implications in designing future therapeutic measures against several diseases like cancer and other things that you mentioned. Thank you all for being present, and with this we come to a conclusion. And I don't know, and now I hand over to Mamtaji.
the lectures that have been taking place, and I would also like to thank the chairs of the session. And taking this opportunity, I would like to invite Professor Rajagopal Raman and Professor Mallikarjun Chakara to please felicitate the chairs of this session.
नहीं अच्छे से ले लो, चारों को लेके।
चारों को नहीं, वो अलग-अलग
Please.
Thank you.